Well, welcome everybody. It's a great crowd, and it's great to see all of you here. This is the first Dean's Lecture of the year of 2020, and it's terrific to have everybody here. As you know, these Dean's Lectures are very special occasions, not just for the individual who is being honored and asked to give the Dean's Lecture, but for all of us as a community here at the school, because it gives us an opportunity to hear from an outstanding group of newly promoted and appointed faculty. And this session today will be no exception. Achieving the appointment of professor represents incredible accomplishments by our faculty and confirms not only the respect that we share for this individual, but for the respect that the individual has garnered throughout the country and globally. Today's event will draw attention to the remarkable work of Dr. Hongkai Ji, and it is my pleasure to introduce him. The central theme of Dr. Ji's research is developing statistical and computational models for analyzing big and complex data, and in particular, high throughput genomic data. He develops tools and then applies these tools to study gene regulatory programs in development and diseases. And I was just chatting with him before and going through his CV. I was just amazed at how many people he currently collaborates with or has collaborated with in the past. And he is the quintessential collaborator, working with people across the school and indeed across the university. And it's very impressive. In 2007, Dr. Ji came to the Bloomberg School as an assistant professor in biostatistics. He was then promoted to associate professor in 2013 and to full professor in 2018. He has served as associate chair of biostatistics since 2018 and also has been that department's graduate program director since 2017. He's affiliated as a faculty member with the Institute for Data-Intensive Engineering and Sciences, the High Throughput Biology Center, and the Center for Computational Biology. Reflecting his stature in the field, Dr. Ji is associate editor for statistical applications in genetics and molecular biology, as well as statistics and biosciences. He is also a member of multiple professional societies, as you can imagine, and is currently program chair for the American Statistical Association section on statistics in genomics and genetics. In addition to his intellectual prowess, Dr. Ji is a respected educator and advisor to many, many students over the years, and currently teaches the course of advanced methods in biostatistics. Dr. Ji received a Bachelor of Engineering degree in automation and a Master's of Engineering degree in pattern recognition and artificial intelligence, both from Tsinghua University. He then went on to receive both an MA as well as a PhD in statistics from Harvard University and was a visiting student at Stanford University from 2004 to 2007. Dr. Ji has been the recipient of many awards and honors, including a Faculty Innovation Award from Johns Hopkins in 2007 and a Johns Hopkins Catalyst Award in 2016, specifically to support the development of computational and statistical tools for comparative functional genomics. In recognitions of all his many accomplishments and contributions to the field, he was elected a fellow of the American Statistical Association just this past year in 2019. It is a huge pleasure and an honor to welcome Professor Ji as today's Dean's Lecturer, and please join me in welcoming him.
Okay, thanks, Adam, for the nice introduction. And before I start, I would like to thank all my colleagues and friends from the Department of Biostatistics, and I'm grateful for your support throughout all these years, ever since I joined the department. And thank you very much for making this department such a wonderful place. And honestly, I cannot think of a better place for me to grow from assistant professor to a full professor. And similarly, I'm also grateful for the support provided by the school and by the university. And of course, I want to say thank you to all my lab members and collaborators. And it's a great honor to have had the opportunity to work together with you to make our world better. Today, I'm going to talk about single-cell genomics. And first, what is single-cell genomics? And simply put, single-cell genomics is a field where people trying to use high-throughput genomic technologies to analyze individual cells. And when biologists analyze a biological sample, usually the sample contains many, many cells. And these cells can come from many different cell types, and they may have different cell states. Traditional technologies such as RNA-seq, ChIP-seq, and microarrays are bulk technologies. And when you analyze the sample using these technologies, you are trying to analyze the aggregated behavior of all cells. For example, if you want to measure gene expression, you first extract transcript from all cells and put them together and analyze using RNA-seq. And as a result, you get the expression value for each and every gene, which can be organized into a vector for each sample. So this is the data, what you get. And a limitation of this technology is that if there are important biological signals carried by a small subset of cells, you may not be able to see it because those signals may be buried in the aggregated signal. Unlike the bulk technologies, single-cell technologies try to analyze each individual cell. Again, if you want to measure gene expression using, say, single-cell RNA sequencing, what you get is the activity of each individual gene in each individual cell. As a result, you have a matrix where rows are genes and columns are cells. And with the ability to measure each individual cell, now you can discover previously unknown cell subpopulations and also identify important biological signals that are carried by a small subset of cells. And many of these signals may help you tell the differences between, say, disease samples and the normal samples. Single-cell genomic technologies are relatively new, but these technologies evolve really fast. The first single-cell RNA sequencing study was published in 2009. Since then, many new technologies have been developed to measure different aspects of a single cell. Examples include transcriptome, epigenome, chromatin interactions, etc., etc. And these technologies are rapidly transforming biomedical research. In 2013, single-cell sequencing was selected by the Nature Methods journal as the method of the year. And just a month ago, in January of 2020, Nature Methods once again selected the single-cell multimodal omics as the method of the year. Applications of single-cell genomic technologies are exploding. In addition to using these technologies to discover novel cell types, people have also been using these technologies to study cells' developmental lineages, to map genes' spatial expression patterns, to study tumor microenvironment and cell-cell interactions. And these are only a few examples. Leveraging the unique power of single-cell technologies, the Human Cell Atlas project proposes to create a comprehensive reference map of all human cells and use it as the basis for understanding human health and disease. Here at Hopkins, single-cell genomic research is also quickly expanding. Many investigators have started to generate single-cell genomic data, and now we already have a big single-cell community at the university. And just 2 weeks ago, School of Medicine Vice Dean for Research organized a meeting to discuss an institutional single-cell RNA-seq initiative. And more than 20 faculty members attended that meeting or participated in the discussion. And this is only an incomplete list of investigators who are using single-cell genomics. And there are many other investigators across the campus who are generating single-cell genomic data, although they did not attend the meeting. Perhaps my own experience with single-cell genomics provides a good example to show the rapid growth of single-cell genomic research. And we started to look at some of the public available single-cell genomic data in 2015. And in 2016, we published our first single-cell methods paper. And around that time, we were trying to look for local collaborators who can generate single-cell genomic data. And we did not find many people at that time. And the first local single-cell data we obtained actually was from Andy Feinberg's lab. And then after that, we gradually developed a collaboration with Dong Zhak from the Wilmer's Eye Institute, and another collaboration with a team led by Zhu Pardo at the Bloomberg Chemo Institute for Cancer Immunotherapy. And these slowly evolved over a period of 3 years. And then suddenly the situation changed after 2019. And within just 1 year, we received a large number of requests for collaboration in terms of analyzing single-cell genomic data. You can see these projects cover many different topics from HIV to vaccine and others. And clearly, we feel that the trend is changing. 4 or 5 years ago, we were looking for data, and now data are trying to find us. Obviously, there's a reason that data are looking for us. The reason is it is not easy to analyze single-cell genomic data. There are many challenges. First, data is big. For example, for this collaborative project, we are analyzing a data that consists of more than 1 billion sequence reads, trying to measure gene expression levels for about 20,000 genes in more than 300,000 cells. And just the raw sequence data occupies more than 5 terabytes disk space. Another data set, similarly, more than 1 million cells, and the raw data, more than 6 terabytes disk space. And this type of data becomes a norm of our collaborative single-cell genomic project. And data size is not the only challenge. There are many other challenges that are equally challenging and perhaps even more challenging. For example, we have to deal with new data structures. We have to deal with different kinds of technical noises and biases in the data. We have to deal with the data sparsity due to assay of small amount of molecular materials. And we also have to integrate many different data types. Because of all these challenges, today, data science and statistics has become a crucial component for single-cell genomic studies. In my talk today, I'm going to share some of our past and ongoing experiences in single-cell genomic studies. And the goal or the hope is to somehow present some of these challenges and opportunities single-cell genomics brings to data science and statistics, and discuss how data science and statistics can improve single-cell genomic studies, and what we as data scientists or statisticians can bring to the table. And I'm also going to discuss how we can combine single-cell genomic technologies with data science and statistics to study interesting problems in basic biology and diseases, including how to analyze patient samples. And all these examples and stories that I'm going to share today is also meant to provide a context for this audience to start thinking about what it means for public health, and how we can leverage this transformative technology to advance public health research. All right, first, we know single-cell genomics creates a lot of challenges, but for statisticians and for data scientists, these are also opportunities. Let me use one example to illustrate that. And let's first consider this example. Here, we are trying to analyze the bone marrow single-cell RNA-seq data generated by Human Cell Atlas. In this plot, each dot represents a cell, and each cell corresponds to a vector that consists of expression values of thousands to tens of thousands of genes. And the cells are projected to a low-dimensional space using the first 2 principal components. When you look at this data, what do you see? Perhaps you see a star-shaped pattern with 3 branches, right? And you may ask, what is this pattern? What does it mean? What are these 3 branches? It turns out that after people analyze this data and look at the data retrospectively, these branches represent biology. Indeed, in the middle, cells in the middle correspond to the hematopoietic stem cells. And we know this type of stem cells can differentiate into 3 main lineages, including lymphoid lineage, which basically are immune cells, erythroid lineage, which are red blood cells, and myeloid lineage. Okay? And these 3 branches correspond to these 3 differentiation lineages. In other words, the shape you have observed represents some real biology. And we are fortunate that this is a system for which people have a lot of knowledge. However, in many other applications, the biological system in question is poorly studied, and people almost know nothing about the underlying biology. And the question of interest is, can we use the observed data to infer those unobserved underlying biological processes? In 2014, Cole Trapnell's group developed a very interesting method called monocle to address this problem. In their method, they tried to construct a minimal spanning tree to connect all the cells. In this tree, each node represents a cell, and the edges are used to connect different cells. And this tree will connect all the cells. The length of each edge characterizes the similarity of 2 cells. If 2 cells are more similar, their distance will be smaller, so the edge will be shorter. And the monocle will find a spanning tree that connects all the cells, but with the minimal total edge length. And these authors show that if you construct a minimal spanning tree in this way, often this tree captures the biological process. For example, if you move from one end of this trajectory to the other end of the trajectory, observe that the cells actually are progressively changing their states from the undifferentiated states to a differentiated states. And we know in many biological differentiation processes, cells can differentiate into multiple different lineages, creating a branching structure. Because this is a tree, it actually can capture the branching structure. And after you construct the minimal spanning tree, you can assign a pseudo-time for each cell based on their order on this trajectory. And then this will create a pseudo-temporal trajectory or pseudo-time course. And after you get the pseudo-time course, you can then study expression level of each individual gene along the pseudo-time. And when you look at many genes simultaneously, you will then get the information about which genes are activated first, which genes are secondary, which genes are downstream. So you get an understanding of the underlying biological process. It's a very cool method. And however, when we apply this method in some of our data sets, we realize that this method also has a big problem. And the big problem is when your cell number is big, and when the tree structure is complex, often the minimal spanning tree will not accurately capture the underlying biology. For example, in this slide, we show analysis of a bone marrow sample, and the red curves here shows the 2 trajectories running from stem cell to erythroid and to myeloid lineage. And then this plot shows the expression level of a gene, HBA2, along the differentiation trajectory from stem cell to erythroid. And this gene is a known marker gene for red blood cells. So you should expect that the expression level of this gene will increase as cells differentiate. However, when you look at the expression pattern reconstructed using monocle, you see a weird pattern. The gene's expression first increases and then decreases. So it does not accurately capture the biology. Why? A fundamental issue of this minimal spanning tree approach is actually associated with the complexity of the tree space. When the cell number is large, the number of possible spanning trees is huge. And when you are trying to find the optimal spanning tree, you actually are doing an inference associated with very high variability, especially when there is huge amount of technical noises. Okay? And here I'm showing you a toy example. In this example, each node represents a cell. Okay? And then let's assume the true biological time runs from top to bottom. And in this example, the minimal spanning tree solution is not unique. Both A and B shows you a minimal spanning tree. So you at least have 2 minimal spanning trees with equal branch length. And the tree in A does not capture the real biology well because the time runs down and then goes back up. And the tree in B roughly captures the true biological time. However, in the data, there are all kinds of technical noises. When there's a measurement noise that moves the cell slightly, for example, moving this green cell slightly to another place, you can easily check that this tree will no longer be the minimal spanning tree. So if you are trying to find the minimal spanning tree, you are going to find A, which is not the correct solution. Okay, so that is the fundamental problem of minimal spanning tree. Essentially, it's caused by the high variability. And go back to our example. So to see what should be the right direction of pseudo-time trajectory, and here we are showing you the cells with high expression of that HBA2 gene. And as you can see, based on the expression, the ideal direction of the pseudo-temporal trajectory should go from here straight to there instead of having this downward curve. Okay, this downward curve is caused by the variability problem I have mentioned. All right, so here's the problem. How can we solve the problem? I'm lucky enough to have a very talented student, Jason Ji, and he's a PhD student, and we work together to figure out the solution. And we realized that spanning tree is not a typical data structure we see in standard statistical textbooks. However, we also realized that we can borrow some principles in statistical literature. In particular, one principle we can borrow is the bias-variance trade-off, which is very useful for handling problems where you have inaccurate estimates and then you want to improve that estimate. Now the question is, how are you going to do this bias-variance trade-off on a spanning tree? New problem, right? It turns out that there is a simple solution. The solution is, first, let's group similar cells into clusters. So if we group similar cells into clusters and use the cluster center as a node to construct the minimal spanning tree, in this toy example, you can easily fix the problem. And based on this idea, Jason has developed a method called T-scan. And in this approach, again, use the bone marrow example to illustrate. We first cluster cells into different clusters, and then we construct a spanning tree to connect the cluster centers. And compared to the spanning tree you obtained from monocle, you can see the tree is much more stable. After you construct this minimal spanning tree, you can then project each cell onto the tree to derive its pseudo-time. Okay? Simple approach, and it addresses the problem. Now, if you compare the pattern reconstructed using monocle and the pattern reconstructed using T-scan, you can see that this approach fixes the problem you have seen. It correctly recovers the biology. And this is just an example, and indeed we have systematically benchmarked the method, which I will not talk in detail, but there is a third-party benchmark study, which compared many different pseudo-time construction methods. And we're happy to see that T-scan is one of the top performers. And in this plot, what they show is on the x-axis is publication time, on the y-axis is the performance. And T-scan was published in 2016. You can see like after publication, it was a top performer for a long time and performed better than many methods that are developed later. And then later, there's another method called slingshot that outperformed T-scan. And slingshot is a method developed by Sandrine Dudut from the UC Berkeley group. And we actually are happy to see they are outperforming us. Why? Because when they built slingshot, they used the idea of cluster-based minimal spanning tree as the first step, and then add a principal curve fitting as an additional step to improve the method. So we are glad to see that this basic idea of regularizing minimal spanning tree using clustering has become a basic building block for these top-performing methods. And the pseudo-time analysis has many applications. And for example, we have applied the pseudo-time analysis to analyze transcriptional network in T cell fate decision. And we are also collaborating with Andy Feinberg to analyze cells' response to drug treatment in a pancreatic tumor model. And again, in the interest of time, I'm not going to go into the detail of these applications. But the take-home message of this first example is that single-cell genomics provides a lot of challenges, but these challenges also means opportunities for statisticians to develop new methods. In this particular example, you can view the spanning trees as a new way, a new low-dimensional representation of the high-dimensional data. And as a comparison, similar methods we had before are principal component analysis, which uses orthogonal basis to represent high-dimensional data. And there are also principal curve analysis, which models the nonlinear patterns of the data, but does not allow branches. And the minimal spanning tree offers a low-dimensional representation, which allows you to have branches to describe the high-dimensional data. And then in order to handle the complex tree space, you need to do regularization. So that's the contribution as statistician we make to the field. And obviously, this new method also benefits single-cell genomics because you obtain the improved analysis performance. All right, so in this example, we had the data first. And then we had a problem. We want to solve the problem, so we developed a new method. However, data science and statistics actually can offer more than that. And we actually can play a role even before you collect data. And as another example, let's consider the spatial mapping of gene expression. We have talked about the temporal patterns of genes. Spatial patterns of genes are also very important. For example, if you want to study development, and the different genes will express at different locations in an embryo. And the correct spatial expression is crucial for the normal development of an embryo. And abnormal expression may result in many different diseases. Okay. Previously, in order to study gene spatial expression pattern, people have developed many low-throughput technologies. For example, in situ hybridization, which are the images you have seen here. And I previously collaborated with a developmental biologist, and they are generating these in situ images. And it is quite tedious. And there are more than 20,000 genes in the genome. If you want to analyze all the genes, you have to do this experiment for repeat this for 20,000 or more times, and which is almost impossible. And the question is, can we establish a spatial mapping of all genes? Okay, and with single-cell genomic technologies, now it becomes possible. So this is the idea. Okay, obviously we cannot do in situ because that's too low throughput. On the other hand, we can also not survey all the cells because there are too many cells to survey, and the single-cell genomic experiment is also not cheap. And if you want to do all the cells, you probably don't have money. So what do we do? As statistician, a natural idea is, why don't we sample? We can sample a representative set of cells, right? So this is the idea. You could dissociate the tissue that you want to study and homogenize the tissue, and then randomly sample some cells and analyze these cells using single-cell RNA-seq. After this analysis, you get gene expression level for all genes for these cells. And then if you can map these cells back to their original location, then you essentially will have a spatial statistical problem. You have sampled many data points. For each data point, you have measurement. Then you can apply any spatial statistical method to reconstruct the spatial expression landscape for each and every gene, right? So simple sampling idea, but useful for mapping genes' spatial expression. And there's a remaining problem. The remaining problem is we actually don't know the location of the cells, because when you shear these cells, their location information are lost. You don't know where these cells come from. And a solution is maybe we can generate some in situ images for a small number of landmark genes. If we have that information, we can use them as a landmark to decide the location of cells. Right? So you can compare the expression levels in single-cell RNA-seq with the expression level measured in the images to locate these cells. And once you locate these cells, you can then apply spatial statistics to reconstruct all this spatial landscape for tens of thousands of genes. So this just illustrates using the simple sampling idea combined with single-cell genomic technology, you actually can achieve something that previous technology cannot achieve. Another example, and in this example, we are interested in studying gene regulation. And you have already seen that gene expression can change temporally and spatially. And the important question is, how are these gene expression activities controlled? And why do they have these temporal and spatial specific gene expression patterns? And gene expression can be controlled using many mechanisms. One important mechanism is transcriptional regulation, in which transcription factors bind to cis-regulated DNA sequence element to turn on or turn off the nearby genes. And therefore, a crucial step to understand the gene regulation is to systematically measure this activity of cis-regulated element, or the activity bound by transcription factors. And this genome-wide analysis of regulated element activities is called regulome mapping. So if you have the regulome information combined with gene expression information, hopefully you could have a better understanding of why a gene expressed at a specific time and a specific location. Obviously, RNA-seq cannot directly measure the regulome. And however, people have developed many other technologies to measure the regulome. Examples include ChIP-seq, DNA-seq, and ATAC-seq. And these are all bulk technologies. And more recently, people have developed single-cell regulome mapping technologies, such as single-cell ATAC-seq. And when you look at the single-cell ATAC-seq as an example, you realize that the single-cell regulome technology also has its limitations. And a typical single-cell ATAC-seq dataset contains thousands of cells, and for each cell, you have tens of thousands of sequence reads. However, as a comparison, you have millions of regulated elements in the genome. So on average, most of the cis-regulated elements will not receive any read. And for the remaining regulated element, at most they receive 1 or 2 reads, and you seldom see more than 2 reads. There's also a fundamental reason here, because here you're trying to analyze DNA, and within a single cell, there are only 2 copies of DNA that's assayable. And another limitation of single-cell ATAC-seq is that when it analyzes a cell gets destroyed, so you cannot analyze it again. You only get to analyze that cell at one single time point. Okay. As a result, what you get is a highly sparse and discrete data at one time point. It cannot accurately measure the activity of a regulated element. Why? Because in principle, we believe that the regulated element activities is a continuous measure. When you think about the protein DNA binding process, it is actually a temporal stochastic process. At one time point, protein comes here and sits to the DNA, and next time point, they dissociate from each other. And then after a while, the protein comes back and sits again. So it's a temporal stochastic process. The activity, steady state activity of a cis-regulated element should be characterized using the probability that the protein sits there at a random time point. And probability is a continuous measure rather than a discrete number 0, 1, or 2. Therefore, the current technology cannot capture that real continuous activity of a cis-regulated element. There are more problems. In order to study gene regulation, hopefully you want to have information about both the transcriptome and the regulome. However, single-cell RNA-seq can only measure the transcriptome, but not regulome. And the single-cell ATAC-seq can only measure the regulome, but not transcriptome. And in addition to that, they also have limitations. And both of these technologies will destroy cells. So if you analyze a cell using one technology, you cannot no longer use the same cell for another assay. And in order to get information from the same cell, people are actively developing single-cell multi-omic technologies, and many technologies have already been developed. However, most of these technologies still have many limitations, such as limited throughput and non-trivial protocol. So in short, this multi-omic analysis in single cell is still quite challenging. In parallel, when you observe the publicly available data, you realize that most of the available single-cell genomic data today actually are single-cell RNA-seq data. You don't have corresponding regulome information. So you really have a little amount of data about the regulome. This constrains our ability to study gene regulation. So as a data scientist and statistician, we start to think about what can we do here, right? And it turns out that there's a data science solution. It's based on observation that there are many large projects such as ENCODE, Roadmap Epigenomics, and they have generated many different types of genomic data for a variety of different cell types. And we sort of hypothesize maybe we can leverage this rich amount of publicly available data to train some machine learning models. And if you can train a prediction model that uses gene expression data to predict regulome, then maybe we can solve the problem. We can apply this method to the single-cell RNA-seq data we have. So we decided to try this idea using the bulk technology first. And we have a great research team here, including Wei Qiang and Ben and Fang are two former postdocs working with me. And we have Jason and we have Jia Wei also from Biostat department. And we also have wonderful collaborators from Kennedy Krieger Institute, and including Ming Yao and Yin Chao. Okay, so we tried this idea. Of course, this is also not easy because here we are trying to deal with a high-dimensional prediction problem. And we want to use gene expression, which is a vector consisting of tens of thousands of genes, to predict genome-wide regulome chromatin accessibility, which is a vector consisting of millions of regulatory elements. You can see the scale of this prediction problem. And fortunately, our group has figured out the solution, and it's called BIRD, and representing big data regression for predicting DNase I hypersensitivity. Again, I'm not going to go to the detail, but it worked for the bulk data. And later, Wei Qiang, Jason, and Wei Xiang Fang from my group extended this idea to single-cell RNA-seq. And this shows how it worked for single-cell RNA-seq. In this slide, what you see is a genomic region, and the different tracks shows the chromatin accessibility obtained using different methods. The bottom track is the signal obtained from bulk DNA-seq, and we view it as a gold standard. Okay, and these two tracks are data you obtained from single-cell ATAC-seq. These are state-of-the-art technologies, right? And then, as you can see, because these data are highly sparse, they cannot accurately capture the gold standard. And this track shows BIRD prediction based on single-cell RNA-seq using the same number of cells, and you can see the pattern better captures the gold standard. When you do a systematic evaluation, we found this surprisingly that BIRD prediction consistently outperformed the state-of-the-art single-cell ATAC-seq when you hold the cell number the same. Okay, when the cell number is not big, we consistently outperform the experimental technology. Okay, it's very surprising to us. And with this approach, we can then boost the value of all our single-cell RNA-seq data. For example, if we go back to the bone marrow example, we can construct the minimal spanning tree to analyze the pseudo-time. And now we can also make predictions, and we can predict how each regulatory element change their activities along this pseudo-time. For example, GATA1 is a marker gene for the erythroid lineage. Its gene expression increases during the differentiation. And here, what you see is the predicted regulatory activity that are bound by GATA1, because GATA1 is a transcription factor you can see is also increasing. Okay, so now this shows you that you actually can generate some data without actually doing the regular mapping experiment. All right, so what this example tells you is that actually data science and statistics can also create many new opportunities for single-cell genomics. Actually, this example shows we can create new technologies that you may not have, or you may not, the quality is not good enough. Okay. In my previous examples, you have already seen that how single-cell genomics and data science may be applied to some basic biology problems. And next, I'm also going to show you an example where we are trying to use this method to analyze patient samples. And first, we want to mention a study that involves people from BKI, the Bloomberg Kimmel Institute for Cancer Immunotherapy, including Drew, Kelly, Vasan, Jaja, and Justina, and many people from Biostatistics department also participate in this project, including Jason, Boyang, Wenping, Wei Chang, and Ni. And in this project, we are trying to study patients' response to cancer immunotherapy. So cancer immunotherapy leverages your immune system to treat cancers. So our immune cells can attack bad cells and kill them. This includes the tumor cells. And there is also a checkpoint mechanism that prevents our immune cells to attack normal cells. And tumor cells are smart. And when they evolve, they learn how to utilize that checkpoint to escape the immune attack. For example, some tumor cells can express a ligand called PD-L1. And this ligand can interact with the receptor PD-1 on T cells. And then when this interaction happens, it's like these two cells are shaking hands, and the tumor cell is saying, "Hi, I'm friend, not enemy, don't kill me." And immune cell, the T cell, got tricked, and the cancer cell will escape. Okay, so in immunotherapy, people try to use drugs to block this checkpoint. For example, you can develop antibody to block the PD-1 receptor so that it's no longer recognizing PD-L1, and now immune cells will not get tricked and it will kill the tumor cell. And in non-small cell lung cancer, the immunotherapy has shown to have substantially improved the efficacy compared to standard treatments such as chemo. However, still, there are a large number of patients who do not respond to this type of therapy. In one report, for example, people have reported only 20% of patients actually respond to immunotherapy. And the natural question is, why some people respond, why some people do not respond? Can we find out the molecular mechanisms behind it? Can we use that to develop better intervention methods? And in this context, the BKI conducted a phase 2 clinical trial, and in which they treat resectable non-small cell lung cancer patients using neoadjuvant immunotherapy. So each patient get 2 doses of anti-PD-1 at day 0 and day 14 respectively. And 1 month after the first dose, the patient will go through surgery to get the tumor removed. And during the surgery, some of the normal lung tissue will also get removed. And then patient will get followed up. And the blood samples are collected throughout this time course. And we also have the tumor samples as well as normal lung samples. And then the BKI group has measured many different genomic modalities. And for this talk today, I'm only going to focus on the single-cell genomic part of it. And they have actually used the single-cell RNA-seq and the single-cell T-cell receptor sequencing to measure, to profile the T cells in these tumors, or so-called tumor-infiltrating T cells. Okay, so natural question is, can you tell the difference between responder and non-responder? But this problem itself is already not that easy. And say, for example, you want to compare 2 responders versus 2 non-responders. If you use the traditional bulk technology, this problem amounts to a t-test-like problem. You have 2 measurements from the responder group, 2 measurements from the non-responder group for each gene. And then you can perform a t-test to see whether it's differential expression or not. But for single-cell data, instead of having 1 measurement for each sample, you have measurement for many cells. So you are comparing different groups of cells, okay? So what makes this more complicated is that these cells are heterogeneous. They have different cell types. And we only want to compare the same cell type across samples. For example, we want to compare CD8 cells versus CD8 cells. We want to compare CD4 cells versus CD4 cells. And we don't want to compare apples with oranges. And when you have the data, you don't know what's the cell type for each cell. So you have to figure out which cells in different samples represent the same cell type, okay? So you have to identify cells of the same cell type and then compare the expression level. Even after you identify the same cells from the same cell type, the data structure is also complex because you have multiple levels of heterogeneity. You have patient-to-patient variability. You also have cell-to-cell variability within each patient. So you can see that data is complex. And fortunately, we have an outstanding group of students and postdocs, and then they have figured out the solution, and they develop a pipeline that aligns cells across different patients, and then perform differential analysis using this data through some generalized linear mix models, plus some advanced techniques to handle the small sample size and technical noise and many things, okay? And then after this development of these pipelines, they have systematically analyzed data and constructed an integrated T-cell atlas using this patient data. And this project is still ongoing, but initial analysis shows that there are some promising signatures. For example, some genes are expressed in tumors of non-responder patients who do not respond to the therapy, and untreated patient, and they do not show up in the responders. And Wei Qiang further applied the BIRD method we have developed to systematically predict genome-wide regulatory element activities for each patient sample. And again, you can see that responders and non-responders, they show some clear differences. So now we are trying to further understand what are the biology behind it, and hopefully in the future, if these signatures can be repeated, say, in peripheral blood, maybe before the surgery, you can predict who will respond, who will not respond, and that will have some translational potential. And here is another example of patient data. In this example, we again collaborate with Zhu and also Christina and Chetan from School of Medicine. And here, they have collected single-cell RNA-seq data for glioblastoma, which is a type of brain tumor. And they want to understand the cell origins of this tumor. And they hypothesize maybe different origins of the tumor may correspond to different outcome. Okay. And to figure out the origins of this tumor, Wenping Ho and Jason from my group has collected a number of published single-cell RNA-seq data set for brains. And they constructed a systematic cell atlas for brain. And they applied the pseudo-time analysis to construct the developmental process in which neural stem cells differentiate into astrocytes, oligodendrocytes, and neurons. Okay, so we recovered the developmental lineage. And then they projected each patient sample to this reference lineage. And it turns out that different patients, the cells are distributed differently. Some patient cells are distributed here, some patient cells are distributed there. And now we are trying to figure out whether these different cell distributions on this reference atlas has anything to do with their outcome. All right, so these are the examples, ongoing research that involve patient. So at this point, you have seen that single-cell genomics and data science, by combining them, how we can combine them to address some basic biology and clinical study problems. Then how about public health? When you look at my collaborators, you can see most of them actually are from School of Medicine. However, in the most recent year, 2019, we have a new collaborator sitting here, Diane, who's from public health. And Diane is interested in study immune response to measles. And then her group has generated single-cell RNA-seq data using monkey models. And then now we are collaborating on analyzing those data. And you can see that the public health start to pick up this new technology. And indeed, in our school, there are already many people who are working on single-cell genomics. For example, on the single-cell genomic data science and statistics part, we have, in our department, we have multiple faculty members working on this, Stephanie, Casper, and Ni, Wei Chang, and me. And we work on different things. And Stephanie and Casper actually are a member of Human Cell Atlas. And in terms of data generation, we also have people in this school who are generating single-cell data, including Diane, and I also talked with Gio Wang, and he's also generating some data. And we have submitted a sidebar proposal led by Anthony Liang. In that proposal, we propose to use single-cell genomic technology to study immune response to alpha virus. Okay, so this is just to show that the things are happening here at public health. However, I think we haven't achieved the full potential of this technology yet. And I think there are many unique opportunities that we can explore at this school. For example, I'm lucky to have collaborations with some people who have large cohort. And one of my collaborators is Professor Xiaobing Wang, and she has a wonderful Boston birth cohort in which she collects paired mother and infant data and monitor the child growth over time. So they have this longitudinal cohort, and they have blood samples along this time. And there are many similar cohorts, I guess, in this school. And the natural question is, can we leverage the transformative single-cell genomic technology to analyze this large cohort data? Of course, cost is a consideration. Currently, the cost of a single-cell genomic experiment is still high. But I'm optimistic because when you look back 10 years ago, 20 years ago, when people just have the microarray technology or sequencing technology, their cost is also high. And the people at that time don't think that they can be applied to large cohort. But then a few years later, you can see all these large-scale studies where you apply microarrays or high-throughput sequencing to study large cohort. And I think single-cell genomics is in a similar situation. Now, the cost is a little bit high, but in the next couple of years, maybe the cost will reduce to a level that you can apply to a large cohort. So if you can analyze large cohort using this data, for example, you can address many interesting public health questions. Say, if you analyze this Boston birth cohort, you probably can identify molecular signatures in your immune system in the early childhood or in mother's blood that may predict the disease outcome of later development of a child, for example, asthma, food allergy, and other things. So we, as public health school, we have unique advantage. We have all these big cohorts. And I think it's really a good time for us to start thinking about what this transformative technology means for us and how can we best use this technology to advance public health research. The real problem now is, are you ready for this new challenge? With that, I'm going to conclude my talk. Thank you very much.
Well, that was great, Hongkai. Thank you. It was incredibly well-delivered talk, and we thank you very much for this. Questions? There we go. Questions? There must be questions. Yes, Karen.
So Hongkai, that was terrific. In the first part of your talk about spanning trees,
of course, to me, it sounded like a latent variable model. You have an underlying biology, you have a manifestation, you have problems of identifiability, which to others in the crowd means exactly what you said, that different models essentially explain the data equally well. The data can't distinguish between them. In your case, it sounded like the biology was pretty definitive, such that you could distinguish the correct model from the poor one. But I could imagine there might be other situations where the biology is less known, and you could end up with competing representations of the data, which then would need to go back to somebody's lab to do a validation experiment or something. Are you finding that as well, or is it usually you have the biology worked out? And yeah.
Thank you. That's a brilliant question. Actually, you're proposing a research direction. and yes, in many real applications, you do see that even with the most advanced spanning tree approaches, the spanning tree is still probably is not correct, and there are competing spanning trees. So a open problem in this field is how are you going to characterize the uncertainty of spanning trees? And all I have talked about is to provide a point estimate of the tree, right? So you can imagine their variability, and if you can correctly characterize the variability, hopefully that can partially address your question. If two trees, based on their variability, are not significantly different, then maybe you will have a different interpretation compared to a case where the two trees have significant difference. So suppose two trees have significant difference, then you have to work with your biological collaborator to do some biological experiment to ascertain which tree is the right tree. Yeah.
Yes, Scott.
I want to try to draw an analogy. Imagine the data are instead the presence or absence of each of 20,000 different billing codes. And that's sort of the outcome. It's like the RNA product. And then there are these regulatory moments where a diagnosis is made, or in the clinical record, there's some information that regulates what sorts of services will be provided and bills generated. And the people are traveling through time. And I wonder if there's some opportunity to take some of what you've developed and apply it to an entirely different problem with the similar sort of insights that you generate.
Well, that's a very good question. And honestly, I haven't looked at the type of data you have asked about, so I don't have a specific idea how each of these specific measures I mentioned here can be directly applied or not. But I'm pretty sure many of these general principles may be applied. For example, maybe not directly address your question, but for example, the minimal spanning trees, the tree idea, you can imagine if you have a population and you want to study, so if this human population have different disease developmental trajectories, and then you collect their epidemiology data or EHR data or medical image data or like fMRI data, right? And then you can similarly treat each patient as a node in the tree and construct a minimal spanning tree type of representation of the data to study the progressive changes of disease states, and then to analyze how features gradually change along the tree. And then for other types of new data structure, perhaps you can adapt the variance bias trade-off ideas to specific design approach to handle those data sets. And then for prediction problems, for the high dimensional prediction problems, and obviously also it depends on the specific data structure you have. And you can use, say, a state-of-the-art deep learning method if you really have a huge sample size, or if you have a small sample size, you probably need to use some methods like we have developed here for BIRD for predicting the regulums. So not specific answer to your question, but hope is towards that direction.
Jeff.
As always, that was a brilliant talk, Hongkai. I thought the example with the non-responders and the untreated, the differential expression result was really compelling. Do you think that this kind of single-cell technology is going to get used more and more in these sort of diagnosing where immunotherapy works and doesn't work? And have you started to think about how the results you've found could be used to take the next step in terms of therapy assignment beyond the first neoadjuvant therapy?
Yeah, that's a very good question. So for now, single-cell technology is still used to discover signals. So for example, in the plot you mentioned here, so you found that there's a small cell subpopulation in which a gene is highly expressed in non-responders and untreated patients, but low in responders. And then using this expensive experiment, we can identify those small cell populations. We can know what are those cell types, and we can know what are these genes. And then in order to translate it into clinics, and perhaps you can develop cheaper assays to specifically analyze that specific cell population and specific gene. And hopefully that will be cheaper. That's one consideration. Another consideration is currently our analysis are based on tumors. So in order to get tumors, you have to do the surgery. So the tumors get removed. And then ideally, you would like to predict it before treatment, right? So therefore, the natural next step for us is to also analyze the PBMC, the blood samples, before you do the surgery. And if you can identify molecular signatures that correlate with this pattern, and then those signatures will be able to suggest some biomarkers for your prediction, whether a patient will respond or not respond. And if you have that information, then you can consider, say, developing adaptive trials to assign patients based on, say, their sort of different molecular profiles. And then that obviously will be a long-term project.
Hey there. So I'm sorry I walked in a little bit late, so I may not get the details of my question right, but I'm going to try. So in several parts of this, you've given examples where you can apply data science or even study design recommendations. And you've shown places where if you can't sample all cells and all tissues at all developmental points, how you can use reference data to cast or predict onto places where you don't have measured data, right? Yes. And then you ended by saying things like BBC or other cohort studies. Right now, we can't afford to do single cell analyses on everything, and we certainly will never be able really to afford all cells, all tissues, all times. So putting those together, have you thought about how you can help us, cohort PIs and epidemiologists to think through smart designs where if we wanted to add single cell components, knowing that we can't do it in all things, where there might be available referencing data that would be most useful and optimal to do hybrid, where we're doing bulk tissue kinds of studies as well as single cell at the same time? That's a great question. And that's right on point. And on top of my head, for example, there are things we can do. Say you have a large cohort, and if you want to study the time course over the lifespan, you obviously cannot sample all the time point. But maybe in statistics, we can first do a low frequency sampling several time points from a small cohort and see how does the pattern generally look like. And then based on whether the curves change dramatically or not, we can design where to put more sampling points, right? And then that's one consideration. And then to fill in the gap between two sampling points, and you are right, and then perhaps we can use the prediction ideas to predict things in between. And hope that answers your question.
My question is about BIRD. So after you've imputed elements of the regulome based on gene expression data, presumably you want to do, for example, differential chromatin accessibility or some sort of comparison. My question is, how is that comparison, are the techniques for those comparisons mature, and would I trust a comparison based on the output of BIRD? Does the uncertainty in the imputation get carried across into a differential, for example, chromatin accessibility analysis? And is there enough power to actually do differential regulome stuff based on the output of BERT?
Again, a great question. And honestly, we haven't started to work on that problem yet. So currently, after we do prediction, when we analyze differential signals, we just compute the point estimate. And because we know that the variance you compute based on the predicted data perhaps does not fully capture the real variability. And then we haven't have a solution yet. So we are trying to think about that problem and work out a solution. And if you're interested, you can also contribute to this study.
Yes. From a biology perspective, gene expression is not only translated into protein expression. So I'm wondering if currently there is any method to measure protein expression at a single cell level, and like what is your opinion on combining different type of information like gene expression and protein to make prediction?
Yeah, measuring proteins are more difficult, but there are some technologies to measure single cell proteins, maybe at a lower throughput. And then, for example, there's some technology that can measure the surface markers of cells, say immune cells, and then in conjunction with the RNA-seq or like other genomic modalities. So it's possible, and the technology are still developing, and it's still not mature enough. This is the current states. So.
Well, Hongkai, it was a brilliant presentation, brilliant work, lots of tough questions that you really handled extremely well. There's so much possibility here. So congratulations on all the work you're doing, and keep doing it. So.
Thank you.
At this point, I'd invite everybody to a reception out by the Wall of Wonder. And thanks for coming.